Hi, I am Pradeep from Newcastle University, UK. Auditory working memory pertains to the process of keeping sounds in mind for short durations when the sounds are not present in the environment. Our previous fMRI study in humans with a maintenance delay of 16 seconds showed enhanced bold activity in a network consisting of auditory cortex, hippocampus, and inferior frontal gyrus. This work aims to reveal the spectral and temporal dynamics underlying the neuronal activity during maintenance of pitch of a single tone in humans using MEG. I recorded MEG or scalp magnetic signals in 17 subjects while they maintained one of the two presented tones or ignored both the tones in the control condition. After 12 seconds, they compared the pitch of a test tone with the tone maintained. Evoked responses showed persistent activity throughout retention compared to the pre-stimulus baseline, but activity only at the start of retention compared to the control condition. This evoked activity was source localized to the auditory cortex. Induced responses during the first second of retention showed enhanced theta in the medial prefrontal cortex, suppressed alpha in the left auditory cortex, and enhanced beta in the cerebellum. 19 new subjects were presented with a tone and a Gabor patch and a cue indicating whether to maintain auditory or visual information for 12 seconds. Induced responses during the first second of retention in the auditory condition yielded results similar to those observed in the first experiment, namely suppressed alpha in the auditory cortex, enhanced theta in the medial prefrontal cortex, and enhanced beta in the cerebellum. Connectivity analysis showed that the theta activity in the medial prefrontal cortex was phase locked to activity in the left hippocampus and the left auditory cortex. The beta activity in the cerebellum was phase locked to activity in Broca's area, and this phase locking was correlated to subject's task accuracy. So I speculate that auditory cortex retains representations of sounds enabled by memory consolidation via the medial prefrontal and hippocampal network and cohort rehearsal by the Broca's area in tandem with cerebellum. Thank you.